For decades, biology only offered two ways to make twins. Identical twins come from one fertilized egg that splits into two matching copies partway through development. Fraternal twins come from two separate eggs, each fertilized by a different sperm, resulting in two siblings who simply happen to share a birthday. Every twin on record fit into one of those two categories, no exceptions and no gray area, no third option. Then in 2014, doctors in Brisbane, Australia were monitoring a woman's pregnancy and found a set of twins that broke the rule. The pregnancy looked completely ordinary at first. An early scan showed one placenta and what appeared to be a single amniotic sac, the classic signature of identical twins. Doctors expected two genetically matching babies, same sex and same DNA, the usual story. Then came the 14-week scan, and the sonographer noticed something that should not have been possible for identical twins: sharing one placenta. One fetus appeared to be developing male anatomy. The other appeared to be developing female anatomy. Identical twins share the same chromosomes, which means they are always the same sex. So a boy and a girl sharing a single placenta was not supposed to exist on the twin rulebook at all. Professor Nicholas Fisk led the fetal medicine team caring for the mother and twins at Royal Brisbane and Women's Hospital in 2014, and he called it an exceptional case. He was the one who first proposed the actual mechanism, suggesting the mother's egg had likely been fertilized simultaneously by two of the father's sperm before dividing. Fisk, who had since served as Deputy Vice-Chancellor for Research at the University of New South Wales, worked alongside Dr. Gabbett to push the case from a strange ultrasound finding into a full genetic investigation, rather than writing it off as a technical glitch. That investigation relied on a method most people have never heard of, a SNP microarray, which checks tens of thousands of specific genetic markers at once. Researchers analyzed anywhere from 17,000 to over 120,000 markers depending on the specific test, comparing maternal and paternal allele sharing between the twins in fine detail. That level of resolution is what caught the mismatch. A standard prenatal test would have missed it entirely. The medical team ran genetic tests, and the results, eventually published in the New England Journal of Medicine in 2019, confirmed the twins share all of their mother's DNA, exactly what you would expect from a single egg, but they did not share all of their father's DNA. Somehow, two different sets of paternal genetic material had ended up in the two bodies that came from the same egg. Here's where the biology actually gets strange. Each fertilized egg is only supposed to accept one sperm. The moment sperm number 1 gets in, the egg's outer membrane changes chemically, locking the door behind it. This is one of the most tightly guarded processes in human reproduction. It's not a suggestion. But in this case, two sperm cells got through the door before it closed. That eventually has a name: dispermic fertilization, and it happens more often than most people realize. What almost never happens is what came next. Normally, when two sperm fertilize one egg, the resulting cell ends up with too many chromosomes to survive, and the pregnancy fails almost immediately. In this case, though, before the cell divided, it appears to have sorted itself into 3 distinct genetic lines, one purely maternal and 2 separate combinations of maternal and paternal DNA, one from each sperm. That single fertilized structure then split the same way an identical twin pregnancy splits, except this time the 2 resulting embryos were not identical copies of one genome. They were each built from a different mix of the father's genetic contribution. Researchers gave this process a name: sesquizygotic twinning, from Latin for 1 and a half, because these twins sit exactly halfway between identical and fraternal. They share 100% of their mother's DNA, just like identical twins do. But when it comes to their father's contribution, they share somewhere between the 50% typical of ordinary siblings and the 100% typical of identical twins. In this case, the split explained the mismatched development. Enough of the male typical genetic material ended up concentrated in one twin that it drove male physical development, while the other twin developed along a female path. I hope I say this correctly, but here's the detail that makes this case remarkable even among sesquizygotic twins. When two sperm fertilize one egg, the resulting cell carries 3 full sets of chromosomes instead of 2, a condition called triploidy. Triploid embryos are almost universally lethal. They simply do not develop far enough to become a pregnancy anyone notices. Dr. Gabbett's team proposed that before the very first cell division, the embryo sorted its first 3 chromosome sets into separate cell lines, essentially quarantining each sperm's contribution into its own group of cells, then eliminated the imbalanced line and split the 2 remaining genetic combinations into 2 separate twins. Researchers call this heterogoneic assortment, and it may be the only reason a triploid fertilization event survived at all. The genetic testing also produced a precise number. The Brisbane twins shared 78% of their paternal DNA, not the 50% you would expect from ordinary siblings, closer to identical than fraternal, but still clearly split between 2 distinct paternal contributions. The first known case discovered in the United States in 2007 was different. Those twins were not identified before birth. Doctors only investigated after one of the infants was born with ambiguous genitalia, and testing showed that pairs shared closer to 50% mark. 2 confirmed cases, 2 different paternal DNA percentages, both proving the same underlying mechanism. Although again, it is extremely rare for this to actually go to full term. When the Brisbane case was published, it was only the second confirmed instance of sesquizygotic twinning in all of recorded medical history. That is in the entire known population of confirmed sesquizygotic twins on the planet, compared to roughly 1 in every 250 pregnancies producing identical twins, and 1 in every 90 producing fraternal twins in the United States. Here's what actually gets me about this. For your entire life, you have probably assumed that pregnancy tests, ultrasounds, and basic biology had this figured out completely. 2 sperm, 1 egg, pregnancy fails, end of story, nothing left to discover. But this case only got caught because a routine anatomy scan happened to fall at exactly the point where the mismatched development became visible on ultrasound. If those twins had been the same sex, there would have been nothing to notice. No sonographer, double take, and no referral for genetic testing. No publication in a major medical journal. The twins would have grown up as ordinary identical twins, and the actual mechanism that created them would have stayed invisible. Which raises kind of an uncomfortable question. If this only got detected because of a visible sex mismatch at the ultrasound, how many sesquizygotic twins have been born and never caught? Simply because both babies happen to develop along the same genitalia, basically. Today, the Brisbane twins are being followed by researchers as they grow, partly because their case gave science its clearest window yet into a process that may be quietly rewriting the twin rulebook every so often unnoticed, and that would make them about 12 years old right now that I'm filming this. Dr. Gabbett's team noted that with the rise of routine early genetic testing during pregnancy, more sesquizygotic cases may surface in the coming years. Not because the phenomenon is becoming more common, but because we are finally equipped to see it. For decades, every twin on the planet fit into one of 2 boxes, identical or fraternal. Then a 14-week ultrasound in Australia found a door nobody had opened before, and on the other side of it was a 3rd category that had apparently existed all along, quietly hiding inside a rule everyone assumed was absolute. Thanks for watching today, and I'll catch you in the next one.